My name is Vladimir Bochkarev, and we are co-directing Center of Aging Research together with Slava. And actually, I would like to express my credit and appreciation to Roda Alani, who actually started this entire project, and this is her baby in the beginning, and now it's developed very nicely. You know, so many researchers are communicating and collaboration ongoing. So it's really a big question in aging research, how we can extrapolate data obtained on short-lived mammals to long-lived mammals like centenarians and humans. And apparently, mice and rats, they live up to 4 years, and there are needs for a good animal model, which can be used for extrapolating data to humans. And naked mole rats is one of such models. You know, so they live 10 times longer than other rodents, and they are really centenarians in rodent world. And they live in Kenya desert. They make tunnels underground, you know, and with very low sun exposure. And they have incredible cancer resistance. So in terms of skin cancer, there is no single spontaneous skin cancer incidence in these animals, which is absolutely fantastic. So they can survive without oxygen, very hypoxia resistance for 18 minutes, you know, and researchers use isoflurane to not CO2 to sacrifice them in laboratory conditions. So they have a spatial pain sensitivity, and they have anaerobic metabolism. And we established this facility in Animal Science Center in School of Medicine. So you see that they like high temperature, and they sensitive to noise and vibration, and they like very healthy food, water-enriched food. And apparently we did this study on aged naked mole rats, how skin aging and which kind of mechanisms are involved in skin aging. And remarkably, they have upregulation of anti-cancer genes during aging, which might actually underlie their resistance to skin carcinogenesis. And we established also the study with chemical skin carcinogenesis and show it 100% of resistance to cancer development. And Ikra Fatima is here, who is first author of this paper. So we're really looking forward for collaboration with many other labs in both campuses. And I think power of comparative biology is that, you know, to study animals which are naturally resistant to some diseases might bring such important data for extrapolating to humans and develop new types of therapies for human disorders. Thank you so much.
I am Hadi Nia, Faculty of Biomedical Engineering. I study lung at a cellular resolution, functional lung. So we develop a crystal ribcage in my lab where we can image the functional lung basically at the cellular resolution in real time. How do we do that? We basically take the lung outside the body. The lung is basically ventilated. The lung is perfused, so the lung is in near in vivo situation. But then we design our own ribcage that has the same property of the actual ribcage, but transparent. Now we can see the lung basically inside the ribcage functioning, and you can see basically nicely the entire surface of the lung. If you zoom in with a basically appropriate optical microscopy, you can see the air sacs, the alveoli, in real time, in action. You can see those very nicely. You can go to cellular level. You can see the two metastatic tumors here. Basically, the red cells are alveolar macrophage. The cyan one are CD8 positive T cells. You can see all these basically rich dynamics events in a functioning lung at the cellular resolution. So we could demonstrate this for many diseases, including the healthy lung, metastatic tumors, primary tumor, pneumonia, fibrosis, emphysema, basically in the whole organ. You can zoom in, get the cellular resolution. There is so much information you can get, and you can basically control also a lot of the parameters. So not only imaging, but also controllability. You can basically find more information in these papers. But what about aging? We decided to study aging and see what's the effect of aging on disease progression in the lung. Is it on the resident lung environment or on the circulatory factors? So we developed this basically cross-circulation model. The lung is inside crystal ribcage here. It's connected to basically a mouse that is walking. It's basically conscious. You can see the mouse here. The blood is connected, is going through the tube, a couple of pumps, bowel traps, and then the crystal ribcage up here. You can see the lung is functioning, and then the blood goes back to the mouse. So this can go for a basically long term, and the mouse is basically happy. In the middle, we also put some microfluidic chamber so we can count the immune cells that go into the lung and get out. So it's basically in vivo cytometry here. And you can see the lung capillary function and many other dynamics even perfused with the whole blood. Now that basically we have this cross-circulation model, we decided to study aging this way. So we can use the cross-circulation of a mouse that is donating blood to two lungs. One could be young, one could be old. So this mouse has the same blood basically sending into this lung. So we can, if there is any difference between these two, it would be because of the resident factors. Or we can do the other way. We can have two donors, one young, one old. They would be labeled with different colors for their immune cells, let's say, and then they would go inside the lung, they would fight the pathogen at the same time, in real time, and we can differentiate basically the effect of aging on the circulating immune cells recruited in the lung. As a very preliminary data, here you can see neutrophils in old lung. It's basically this versus this. And neutrophils in the young lung, the neutrophils are coming from the same mouse, so the effect of aging is just on the resident ones. And we already are seeing some basically differences in the neutrophil dynamic activity in this case. Thanks so much.